Genetics, the study that deals with heredity. It is genetics. Long time ago, there were two ideas about how we pass on traits from generation to another generation. Pangenes are small particles in the body, that's what they assume, that forms all of the somatic cells, and they are carried, these small particles, into the gametes, and they are going to be transferred by the gametes. Also, somatic cells that acquired some characteristics like big muscles can be transferred to the offspring. Now, who were those people who came up with this? This is Hippocrates, as well as Charles Darwin thought about that pangenes are the ways that we pass on characteristics to next generations. The counter argument with that somatic cells do not pass anything to the gametes, as well as changes in the somatic cells do not influence gametes. So it's not accepted anymore, that theory, this thinking. The other one is the blending idea that says hereditary materials from the parents get mixed to form blending characters in the offsprings. Like red flower and white flower are mixed together, and the offspring of the flower will have pink flower. Well, this is pretty good so far, but also we will talk about the counter arguments. Who were the one, as there are no names to it, but in the 19th century that was accepted as a passing on traits to the offsprings. The counter argument for that is that traits can reappear after skipping some generations. Like with the flowers, we will have pink flowers, but if we mix two pink flowers together, they can produce red and white flowers, and it's not blending anymore. So, what is that? Oh, in 1860s, there was a person, Gregor Mendel, an Australian monk, who really liked the pea plants and had a huge love towards math and statistics. So he used a huge sample size of pea plants and played in quotes with them, so did his experiment on pea plants. Let's talk about first genetics. He studied the heredity is the passing of characteristics from one generation to another, and genetics is the study of the heredity. The person who is doing it is geneticist. Now, in 1866, he published a paper, and he called heritable factor, the genes now we call it, that they are passed from one organism to another. Heritable features are the characteristics such as the color of the seeds, and the variations of the characters are the traits, like what I said, colors of the seeds can be green or yellow, so these are the traits, the green and yellow. And the character was the color of seed. Mendel's experiment. What he did, he picked the pea plants, and he had several of tall pea plants and several of short pea plants in the garden at a different area in the monastery's garden, because he was a monk in Austria. So they were true breeding plants. That means with self-fertilization, they produce offsprings identical to the parents. So they pollinate themselves. The other ones were the short plants. They were also two breeding organisms. He picked one tall plant and a short plant and cross-pollinate the plants. In nature, self-pollination in the pea plants is normal, and what he did, he cut off the immature stamen, the stamen that contains the pollen and, or the sperm, and used that pollen manually transferring to the carpel, that the female reproductive organ of the flower, to fertilize the egg inside of the carpel. So he cross-pollinated that, and all of the plants that came out from that cross-pollination, the seeds, they became tall plants, all of them. There was no short plant. And these are called the hybrid plants because the offspring of two different varieties is called the hybrid plants. The first generation, the parents, became the P generation, and the F1 generation is the offsprings of the parent generation. The two hybrid plants crossed again through cross-pollination, and he noticed that 4 of them, or 3/4 of the plants that came out from this fertilization became tall, and 1/4 of the plants became short. So that's the F2 generation. And let's go with a percent or ratio. 3 out of 4 was tall and 1 out of 4 was short. What he explained, and this is a monohybrid crossing, pointing out that only one character was only changed, and this point in a monohybrid crossing was the size or the tallness of the plant. So the question what he had to answer for, why did the traits of the shortness disappeared in the first generation, and why did the short plants appeared in the second generation? So the explanation, let's use vocabulary terms now. He defined what alleles were. There are alternative heritable factors, or now we call genes, for characters, and the characters are called for characters, and these alternative factors are the alleles. The second one is that 2 alleles make up one trait, 2 alleles for one trait, and identical alleles make up homozygous, and different alleles make up heterozygous organisms. The alleles can be dominant, and we are using capital letter for that, and the alleles can be recessive using lowercase letters. The fourth one is law of segregation. That means that the gametes, as they form, they contain only one allele in each gamete, and when they fuse together to form the zygote, they form a cell with 2 alleles. Let's check how we can explain it, the hereditary, using Punnett square, and we try to explain the F2 generation. So the gametes, the parents are going to be the F1 parents. They are heterozygous because they contain a capital T and lowercase t. And another parent is also a heterozygous. The gametes in a female are going to follow the law of segregation. So one gamete contains the capital T, another gamete contains the lowercase t, same with the gametes in the male. And now you pull down and cross the letters, and you will fill out the table. And what you will see, all of them that contains capital letter will be tall, 3 out of 4, and all of them that contains no capital letter T will be short. So 1/4 is short. Now, these are the phenotypes. Phenotype is the visible characteristic, the character that we can see. The other one is the genotype. that is different from phenotype. This is the genetic makeup of the organism. The genotype and the phenotype, sometimes they are not the same. In the cell, before we just close it, we have two homologous chromosomes, and at one area, the loci is where a specific location of the character on the chromosome is located. Homologous chromosomes can be homozygous with same alleles and heterozygous with different alleles.